welcome to the Gilroy laboratory. Okay, so this is what a research tab looks like. Turn some lights on. Okay, so we have a load of test tubes. So this box, we use this for like shipping things around. As we send a lot of stuff to NASA, and so we use a lot of dry ice to ship stuff that's frozen. This is a chemical extraction hood, so this is where we do experiments with like dangerous chemicals. These solvents in here are not good for your health, so we have this big extraction fan system that takes those gases and gets rid of them so we remain safe. This is where we mix up our chemicals. So we have some really accurate weighing scales, and we also have this thing is a pH machine, so we can make solutions of different pHs. So we have like acids and alkalis and these kind of buffers back here. And so that way, when we make nutrients for organisms to grow on, we can get the pH just right. When we're making these kind of nutrients, these media compositions, we use lots of different chemicals. There's many different ones. And so we have them labeled like alphabetically. So you can go through and kind of search for chitosan. So this is part of a fungus. So you can trick plants into thinking they've been touched or attacked by fungus with that chemical. Or sodium bicarbonate, you probably have some derivative of that in your kitchen. Okay, cool, so that's the chemical area. This is a typical lab bench that one of our undergraduates works at. So we have pipettes. These things are really important for accurately dispensing small volumes of water. And so we use those pipettes and these tips, and then we pipette loads of liquids into like racks that look a bit like this. And this is the foundation of like modern molecular biology, the capacity to mix together small volumes really accurately, combined with what we call PCR. So PCR is the polymerase chain reaction. These are some old PCR machines, and this is the newer version here with a nice touch screen display. And so what this does is it goes on through thermal cycles. These machines, they heat and cool DNA through specific sequences that allows us to amplify DNA, to make copies of it. And so this is like the foundation of cloning of genes and genetic engineering in general. In collaboration with the PCR machines, we have what we call gel electrophoresis. That's this stuff here. So essentially we can then visualize the DNA by adding a dye, fluorescent dye, which is in one of these tubes just up here. So we mix the dye with some gel and we put it in this tank here, and then we apply an electrical current. That's what this machine here is for, and it puts a positive negative a gradient of charge across this particular system, and then the DNA will migrate towards the positive electrode, because it's a positive anode, because it's negatively charged. Okay, cool, so once we've got some DNA, we've cloned it, the next thing that we do is put it into plants. Here you can see some plants that have dried out. These ones are kind of yellow. So these ones have been genetically modified, and now we're harvesting their seeds, so we can look at the effect of the modification really carefully. Sometimes we need to dispose of these samples. So this is some plants, the seeds have been harvested, but because of GMO regulations, we have to make sure that we pressure cook it. So this is what we call an autoclave. And so that pressure cooker allows us to destroy any biological material that we do not like. For example, I like to sterilize all of my equipment using this pressure cooker in order to make sure that microbes don't grow on my experiments. Another example of a lab bench, so this is where one of the PhD students, Ariel, works. You can see there's lots of tubes and dials. This is actual work in progress, so that's really good to see. Okay, cool. There is one last lab to see, which is the next section. Bye for now.